Hello, everyone, and thank you for joining us today to today's webinar. My name is Stephen Edelson, and I'm a proud member of AFHU's Midwest Region and National Boards. I'm honored to welcome you today to this program featuring the esteemed Dr. Raphael Benhamou. Dr. Benhamou's research laboratory is dedicated to exploring the emerging role of RNA as a target at the intersection of chemical biology, medicinal chemistry, and molecular cellular biology. Those are all very fancy ways of really saying what he's doing, and he's only one of 3 labs in the world to be working on small molecule RNA research to both target diseases and to be able to create cures specifically for those diseases in order to cure those diseases. And as I said a moment ago, he's one of only 3 labs in the world. Before jumping in, one of the things I would like to say, because there will be questions later, is that we've received questions about the labs, their work, and what is happening in these labs. Is work slowing down because of the war that's going on in the Middle East? I think Dr. Benhamou can address that, but we're addressing that as a university with our program of We Are One. And what We Are One has been doing has been funding mental health and resilience for both Jews and Arabs, building a shared society and strengthening the southern city of Ofakim, where we have opened up for the first time in the Negev, a school for one of the schools that was destroyed. What We Are One is also doing, in its main purpose, is to fund scholarships for reservists who have to leave for active duty, and when they come back, we are helping them through school. So with that, I would like to introduce Dr. Raphael Benhamou.
Thank you very much, Stephen, for the introduction. Thank you all for joining us today, and also for the opportunity to Yissum and the Hebrew University for organizing this webinar. And as Stephen said, what we are doing is basically we are developing molecules that are targeting disease-associated RNA. But before I'm starting about the heavy research and science, I would like to introduce a little bit about myself and where I'm coming from. So this is not working, so I will work this way. Okay, so a bit about myself. I was born in France, in Paris, and then I moved when I was 18 years old to Tel Aviv. And then after making the army and completing my deserved duty to my country, I started my study, bachelor and license, and also PhD at Tel Aviv University. Then I moved to Scripps Institute. I did my postdoctoral under the supervision of Matthew Disney. And then I moved back in Jerusalem in 2021, and I created my lab. And my lab is basically a lab that is merging 2 huge fields of research, chemistry and biology. So I'm a chemical biologist. What does that mean? Is basically that I'm using chemistry in order to answer biological questions. And this is a very interdisciplinary lab. We are merging different disciplines of chemistry, such as organic chemistry, analytical chemistry, combinatorial chemistry, medicinal chemistry. And all of this chemistry used in order to go to the biology, where we're using different kind of biology, such as biochemistry, cell biology, molecular biology, and genetics. And the goal of the lab is basically to be able to synthesize new molecule that will have a biological effect. But the most important fact of the lab is that we are targeting RNA. And I hope by the end of this webinar, I'll be able to convince you that there are a lot of different opportunities in targeting RNA. And this is a world of infinite potential. So a bit introduction about RNA and where is it located in the genome and our body. So we are all made of DNA, and this is different portion of the DNA of each different cells. And the DNA is transcribed into RNA. So you can see here the different RNA. This is translated into proteins, And the protein are marking the different cells. You will have different expression of protein in different tissues, such as the brain, arms, the muscles, and so on. And when you look at the full genome, so talking about the 100% DNA portion, you will figure out that 90% of the full genome pocket is transcribed into RNA. And when you use, this is the pocket of the RNA, the transcriptome called, you have only 1 to 2% that is coding to protein. And when you look at the proteome, this is a very important fact, there's only 15% of the full proteome, meaning of full different proteins that we have in our body, that are called druggable, meaning that we can synthesize and design a molecule that will affect these specific proteins. And for 100 years, people have been focusing on this tiny pocket of 0.2% of the genome. And what I'm trying to explain to you today, that we have the way and the tools today to expand and to look at these different pockets and how we can target disease by targeting the full opportunity that the genome is offering to us. And just to give you a proportional view of the size, this is the DNA, this is the RNA transcriptome, and this is the tiny pocket of the protein. And this is only 2% of the full genome. So why minimizing ourselves to only this little dot when we can see what are the opportunities at the RNA transcriptome? And I will show you that many RNA are linked to disease, and that's the reason we're targeting RNA. So here you have an overview of a quick search in PubMed. This is a platform that is merging all the different data and the research. And we made RNA disease research like a few months ago, and we saw that there is an exponential increase in the past 20 years of research that are linking between RNA and diseases. And today is known that RNA is causing disease. All of the diseases are linked to RNA: cancer, diabetes, ALS, autism, and so on. And today we'll talk about few of them. The important aspect of RNA is the structure of RNA. For a long time, people have thought that RNA is this very resemble to the DNA that you can see here. This is the double helix of DNA. But the specificity of RNA is its structure. And here you have the DNA. So the DNA is made of 4 different letter nucleotides. And this is the 3D form of the DNA, double helix, that you probably all known and heard about it. And what you look at RNA is also made of 4 nucleotides, 4 different letters, U, A, G, C. But you can see that the 3D structure of the RNA is more complex than unique. When you're looking at proteins, you will have amino acid. That's the building blocks that are using, not nucleotides. And you also have a very specific structure made of protein. And the uniqueness of the RNA is the one that's making us possible in order to design molecule that will be able to meet and to match the pocket inside the RNA. So when you look at RNA, how they are folded, just think about like one strand that you can fold and over until it's stable, that you cannot fold anymore. And you see the different portion of making such of loops or hairpins. These portions can make pockets, and these pockets can be druggable pockets. Here it's the one version of RNA. You can see that the structure, and also calling it the RNA dancing mode. So you can see that the RNA structure is very dynamic, but also highly structured. So you have a 3D structure for each different RNA. And these are made of different substructures, such as stem loops, internal loops, bulges, 3-way junction, 4-way junctions. These are making the secondary structure of the motifs, meaning that they are the short-range interaction between the different RNA places. And you have also tertiary structure motifs that are making long-range interactions, such as pseudonodes, quadruplex, or stem loop bud junctions. And these different RNA are making very unique form of 3D folding. And here I'm giving you a few examples of RNA form. And the thing that I would like to highlight is the differences between the different RNA that you can see here. So the point is getting that each different sequence of RNA is going to fold into a different way. And this 3D folding is the first part of our research. And you can see ribosomal RNA, microRNA, long non-coding RNA, RNA binding protein, SNORRNA, all of these different RNA are linked to diseases. And if we can design a molecule that will be able to target this RNA, then we can also affect the disease by targeting RNA. And many of them are part of calling non-coding RNA, meaning that they are not translated into protein. And a few months ago, the Nobel Prize in Medicine have been awarding this discovery of a non-coding RNA, the microRNA LIN, that is affecting a lot of different processes in the cells. That's been awarded to Victor Ambros and Gary Rifkin. And just to say that this is very high, this is a very high and interesting field that is bringing a lot of attention in the past years. So how's it working? So basically, we're starting with a sequence of RNA that we recognize that is linked to the disease. And we can use different bioinformatic tools in order to fold the RNA. This is the RNA that is folding. And when we look at the RNA folded, you have two different regions. You have these regions with different pockets, and these pockets have a very high probability that a small molecule will bind to it with a small molecule, you can see here. But there are different portions of the RNA that are not highly folded. Usually, this is the coding sequence of the RNA, the one that is making protein. And this is better to target with antisense oligonucleotide or ASOs. They are recognizing the sequence and not the structure. And by driving RNA with different small molecules, we can affect different RNA pathways, such as RNA synthesis, and therefore will affect the protein expression, RNA processing, RNA splicing linked to a lot of different diseases, RNA binding proteins, or also RNA translation. And I would like to just highlight one example before I start, is the treatment of spinal muscular atrophy, where we have two different ways to treat the disease. One is the ASO, antisense oligonucleotides. It's called Spinraza. And this one is the intrathecal injections. Just to show you a little bit of data about it. So this is the sales in 2023, the project sales in 2021, $1.3 billion. And this is the drug for the SMA. 2 years ago, the small molecule was FDA approved. It's called risdiplam. And this is an oral drug. And in 2029, the projected sales are approximately 3 billion. So it's threefold higher than the ASOs. And there are many reasons why the small molecule is better than the ASO approach. First of all, this is the permeability. Since it's a small molecule, it will be capable to reach to the target in the easiest way. The bioavailability in the body and how it's going to be discarded when it's done to be used. Tissue distribution is going to be better than the ASO. That's why the ASO is by injections and the small molecule is by oral distribution. The cost, the synthesis of a small molecule is much cheaper than the one for the ASOs. The small molecule also is more stable and can live on room temperature, while ASOs are stored at minus 20 degrees. And finally, the small molecule do not need any delivery system in order to reach the target, whereas the ASO have to be encapsulated and inside nanobodies, and these nanobodies are going to reach the target itself. So these are the different advantages of small molecule. And in the lab, we are synthesizing small molecule, and we have two different strategies to bind RNA and to affect RNA. The first one is simple binding. So here you can see the small molecule in blue and orange. And this simple binding way, this strategy is basically having the molecule to interact with the RNA and removing the interaction between the RNA and another biomarker molecule. It can be protein, another RNA, or also a DNA. This is called steric blockage. The second approach is targeted degradation. So we'll have molecule that will be conjugated with a warhead, and this warhead will induce the degradation, the specific cleavage of the RNA that is the root of the disease, and we will decrease very specifically the amount of RNA that is causing the disease. So a few words about the two different approaches. So the first one is the traditional approach, where you have binding and inhibition and the steric blockage. So you have the target of interest. You have only one tractable site when you're tying, so you have to have a molecule that will bind to this tractable site. The small molecule inhibition will have to have full capacity to recognize the site, the active site. The molecule will be affecting the target only when it's the occupancy of the pocket is made. So based on the affinity, the compound can be released from the pocket and then can have no effect anymore. And when you think about it, a ratio is one drug molecule to one target. So this is a 1-to-1. And this is one very important aspect of this approach. And this is causing a decrease in clinical benefits. And disease protein can also have resistance and persistence because of targeting always the same target. And the second approach is targeted degradation strategy. So as I told you, now we're tackling and trying to cleave specifically the target that we are looking for. And how does it work? So now we don't care anymore to bind to this tractable site. We can bind anywhere in the target. So we have new opportunities in order to affect the target of interest. We can tag either the protein or the RNA in order to make it labeled as cellular waste and then cleaved by the cells itself and do not have to induce any exogenous cleavage. We also have here an increase in selectivity since we are talking about molecules that are more complex and then we have more selectivity of the molecule. And finally, the most important aspect is that here we have a molecule that will have a catalytic mechanism, meaning that one molecule will be capable to target one target and to bind to it and to cleave it. And when after the cleavage, you will have the degradation and the molecule will be able to target another one. So here the ratio is not 1-to-1 anymore. You will have a ratio 1-to-1,000, 1-to-1,000,000, even in some cases 1-to-1,000,000,000. So we'll reduce the concentration of the drug that you will need in order to achieve the same effect. And here are the two technologies that we are using in the lab. The first one is photolysis targeting camera technology. So we have two compounds, or the compound that's one side of the compound will target the protein, and the second side will ubiquitinating heat ligase. This will form this ubiquitinated protein of interest, and it will be recognized by the proteasome and degraded by the cell in a more healthy way and natural way. The second approach is the RiboTag, ribonuclease targeting camera, the one that I've been mostly involved in. And here you have a molecule that this molecule, you can see here the structure, will be attached to other molecules that will be capable to recognize the RNA. And after recognizing the RNA, you will have the recruitment of ribonuclease. Ribonuclease is capable to cleave RNA specifically, and it will induce RNA degradation in a very specific manner. And here I would like to show you a few different diseases that we have been capable to target with small molecule and have been involved in the past 5 to 6 years. So starting from bacterial disease, cardiovascular disease, virus, cancer, genetic disease, brain disease, such as depression, neurodegenerative, obesity, aging, all of these different diseases have roots in RNA, and we have been capable to synthesize molecules that are affecting the RNA and also alleviating the associated phenotype of the disease. And I will show you today a few examples of it. And here I just want to show you a few of the diseases that I'm talking about, starting from myotonic dystrophy type 2 and type 1 that are caused by a mutation expansion that you can see here, CCUG or CUG. ALS, amyotrophic lateral sclerosis, being capable also to synthesize molecules that are affecting the ALS route in RNA. Or congenital microRNA, these are non-coding RNA, small non-coding RNA that are linked to a lot of cancers. Viral RNA, we'll talk about it's related to SARS-CoV-2. Long non-coding RNA, also we'll talk about it today. It's related to cancer and aging. Messenger RNA related to cancer and the way to affect the protein expression by targeting messenger RNA. And finally, RNA binding proteins that are complexes that are made between a protein and RNA and can affect a lot of different diseases. I will start by talking about oncogenic microRNA. So these microRNA are short non-coding RNA that are linked to different diseases. And it's known today that dysregulation of this different microRNA can have induced a lot of different cancer progression. And here you have a list of different microRNA that are either upregulated or downregulated. And one of them is microRNA 27A that is highly related and overexpressed in breast cancer. And the way we are targeting is basically to find the molecule that will inhibit the expression of this miR27A. By inhibiting this expression, we also cure the breast cancer phenotype. So how we did it, we basically made and synthesized in the lab libraries of compounds. So here you have a library called DNA encoded library. So this is the compound that's binding to the RNA. And all of the compounds are tagged with a DNA tag. And here we synthesize a 73,000 library of small molecules that we incubated with another library of RNA that we would like to target. And this is the 4,000 library members. And at the end, what we got is after 1 hour of incubation, we got approximately 300 million potential interactions screened. So it's very high throughput way to interrogate RNA to find small molecule that will bind to the RNA that we would like to target. And we interestingly found a molecule that is capable to bind to this pocket of the microRNA 27A. This is located here. And this will block the processing of the microRNA that will inhibit the maturation of miR27A and affect the cancer cell phenotype. So I will show you a few data about it. So here is the testing of the compound, and you can see here all different microRNA not been affected with the molecule, but with 1 micromolar dose, we have been capable to significantly decrease only miR27A with this molecule. And to cut a very long story short, just to show you how it's affecting also cancer. So here it's migration, and the purple color that you can see is correlating with high migration. So we want to see that with dosing of our compound, we will reduce the color and reduce therefore the migration. So you can see at 0.1 micromolar and 1 micromolar, a significant decrease of 50% or 70% was observed in the migration pathway, a very important pathway of cancer in cells. Now we'd like to talk about another target. Here it's oncogenic messenger RNA. And the title of this slide is drugging the undruggable. Many proteins are called proteins that are undruggable since there are no pockets to design a small molecule to it. I talked about it a few slides ago. And instead of looking at targeting the protein, the idea is, okay, if we reduce the messenger RNA abundance, we will also reduce the translation of the protein and therefore decrease the disease pathway. So that's the target of this project. So targeting messenger RNA instead of targeting the protein. Few data about the undruggable proteome. So there are more than 85% of the full proteome, meaning all different proteins in our body that are called undruggable. And when you look at the human protein atlas, you can see the reason why. And here you see 13% of the protein that have been launched drug, 25% that are not suitable for drug targets, and 62% are believed to be undruggable because they are not folding in a way that are making a pocket that we can design a molecule there. So the idea is instead of looking at the protein, we look at the RNA, and we found this interesting pocket. But here, as I said, we will look at a different strategy. It's not a binding strategy, but here it's going to be a selective degrading strategy. We'll use the ribotac approach that I presented 5 slides ago, and we will cleave and degrade the RNA, and therefore we reduce the expression of the RNA. And this is the molecule that has been designed. So you have a molecule that can bind to the RNA. You can see here, this is the pocket where it's binding. However, when the molecule is only binding, there is no functionality. And only after making a ribotac, we have been capable to cleave the RNA and therefore reduce the expression of the messenger RNA and the protein. So here is the data. You can see here in green, this is the ribotac approach. So we've been capable to significantly reduce the messenger RNA. This is the binder. The binder has no effect on the messenger RNA itself. And when we look at the protein expression after the messenger RNA, we saw that with a 10 micromolar dose, we significantly decreased the expression of c-Myc, a non-druggable protein. And also, we measured the different cancer cell phenotype inhibition. So you have proliferation and apoptosis. Proliferation is a very important aspect of cancer. The cancer cells know how to proliferate with a high rate. So we would like to see how we can stop this proliferation. So we reduce approximately 70% of the proliferation rate of the breast cancer. And apoptosis is basically we're activating the death of the cell, and here you can see an increase in apoptosis after 2 days of treatment. Now I would like to talk about another disease and another RNA target, and this one is ALS disease. And the disease is basically made because of a mutation called G4C2. And here we would like to see how we can affect the RNA that's causing the disease. Few words about the ALS, amyotrophic lateral sclerosis. The important aspect of the ALS is that between 2 to 4 years is the life expectancy after the you're diagnosed with the disease. And usually your range of onset is between 40 to 70 years to 70 years. And you have different stages of ALS. It's starting from early stage, where you have trouble buttoning your clothes or gripping objects, the muscles are twitching and cramping. Then very quickly, you move to the middle stage ALS, where the muscles become paralyzed, complicated to walk, they have breathing problem. And then the late stage of ALS, you will have mobility extremely limited, you will have to go with a wheelchair, and breathing function becomes severely impaired. And finally, you have the end stage, where you're fully paralyzed, and you need medication to ease your pain and your anxiety. And one example of a very famous person to have been diagnosed with ALS and to die with ALS is Stephen Hawking. And actually, he's the one that lived the longest after being diagnosed with ALS. And the ALS, one origin of the ALS is this RNA that you can see here, and this mutation. This is a G4C2 expansion. You will have G4C2 repeats that have been located in the patients. And you want to see how we can the goal is to affect this C9orf72 mutation to cleave only the disease mutation and not cleaving the healthy processing of the gene. And what we did, we designed molecule and we tested in cells, and we found that in cells we have been capable to reduce the amount of the genes that are harboring the repeat expansion mutation, and also the poly-GP, one of the proteins that is made because of the expansion. So two different kind of cell lines that are disease derivatives. One is induced pluripotent stem cells, and one is the iPSCs. Then we went to in vivo studies. We generated a mouse model, and we did ICV injection, and you can see even in the brain of the mouse, we have been capable to reduce a 50% extent the mutation that is there and not affecting the healthy variant of the gene, meaning that we have been capable to selectively cleave the target that has bringing the disease mutation and not the healthy one. Another project that we had is targeting virus RNA. So you probably heard, all of you, about the SARS-CoV-2 and the pandemic that we have been through 3 years ago. And here basically is using the RNA of the virus itself and see how we can affect it. So here, this is the way we can look at virus RNA, and this is inside of it. So many are trying to target the spike protein or the membrane protein. But what we are looking for in our approach is targeting the RNA, the one that is responsible for the replication of the virus inside the cells. And we find a very interesting pocket inside the RNA of the virus that's located in the alternator helping of the French-tipped element. So you can see here it's present in SARS-CoV-2, but it's lacking in SARS-CoV-1. And we wanted to design a molecule that will be capable to cleave this RNA and then induce the death of the virus itself. So this is what we did. We used also the RiboTaq approach, we found a molecule capable to bind to other RNA that is recruiting the ribonuclease and therefore degrading the RNA. Just a few data about it. You can see here the significant decrease in the RNA of the SARS-CoV-2 here and here, and not affecting the SARS-CoV-1. And here we have been capable to significantly reduce the replication of the virus RNA of SARS-CoV-2. Another ongoing project, actually, it's targeting long non-coding RNA, and this one is related to cancer and aging. And a few words about long non-coding RNA. For long non-coding RNA are made of between 200 nucleotides to 1,000 of nucleotides. And for a long time, they've been considered as a junk part of our genome, but not anymore. 10 years ago or 15 years ago, they actually discovered that the long non-coding are affecting a lot of different diseases and processing inside the cells. And they have been relating to a lot of diseases such as cancer, neurological disorders, diabetes, kidney disease, cardiovascular disease. And all of them, you can see the name of the different long non-coding RNA, such as MALAT1 or Exist. Even one long non-coding is capable to mute a full chromosome that you have. It's called the Exist1. And the power of the long non-coding RNA is under investigation. And every day you see new research, amazing research made to see how the different long non-coding RNA are affecting the biological processes and especially in diseases. And one of them is called TERA, and TERA is related to telomere elongation. And a few words about telomere is basically at the end of the chromosomes, you will have a portion that is responsible of the stability of the cells. So the longer the telomere is going to be, the more stable is going to be the cells, and the shorter it's going to be, the cell will be related to a very quick death. And the long non-coding RNA, actually called TERA, is capable to regulate this telomere length. And this is actually related to cancer, or since you want to see how we can shorten the chromosome of cancer cells in order to make them die. And then also for aging, because we can also regulate the aging of the cells by regulating the telomere. And one way to look at it is there is in TERA, the long non-coding, a form of RNA called G quadruplex. And this is the G quadruplex. You basically have 4 different letters of G that are repeat one after the others and are making these loops that you can see. This is the interaction between the G, and this is the different loops, and they're stacking one to each other. This is how we can represent them. And the idea was to reduce the TERA that is overexpressed in cancer and also in aging. And the way to reduce it is also using the RiboTag approach. So you have a molecule that binds to the TERA and will degrade the TERA itself. So here, a few in vitro, you can see the control TERA has not been affected when you don't have an RNA and the compound itself. And then when you're merging all of them together, you can see significant decrease from the few first seconds of incubation. And this is the quantification. And then we tested in cells. We've been able to see how we can reduce significantly TERA abundance of 2 different types of TERA, the 13q and the 7p, in the U2S cells that's related to the cancer that's length of telomeres. I've been showing you how to decrease all the time RNA, but what about increasing RNA levels? There are many diseases that there are a lack of RNA and we need to increase. And this is the case in LED7, it's a microRNA tumor suppressor, and it's repressed by an RNA binding protein called Lin28. How does it work? You have here the processing of LED7, so this is inside the nucleus, outside the cytoplasm, and you will have the healthy processing of LED7, and it will reduce the translation of many oncogenic proteins. But in cancer cells, and especially in uterus cancer and ovarian cancer, you will have overexpression of this RNA binding protein that will lead to the degradation of the tumor suppressor, the LED7, the non-coding RNA, that will induce the expression of oncogenic messenger RNA such as KRAS, AMP1, and CMYC. The protein is going to be overexpressed, and it will lead to the ovarian cancer and uterus cancer. So the idea is how we can revert back and have maturation of LED7. So here we first designed molecules that have been capable to degrade using the PROTAC approach, the Lean-28, the one that's responsible of the degradation of lead-7. So you can see here the decrease in the expression of the protein. Then we checked how we can have a boost in the tumor suppressor microRNA lead-7. So you can see a boost in blue and gray using the two different molecules above. For lead-7A, C, and D, that's different variants of the microRNA tumor suppressor. And then finally, we checked the phenotype of the cells in cancer. So you can see here the migration. So we want to see how we can inhibit the migration of the cells when we're dosing. So this is untreated. You can see a great migration after 72 hours. Dosing with our compounds, we've been capable to 100% inhibit the cancer progression and migration in vitro. Few of the ongoing projects that we're currently involved into. We're designing targeted degradation for CB1R. CB1R is a cannabinoid receptor that's responsible of cancer and obesity. We're also synthesizing in our lab for the portion of the RNA that are not capable to be bind with small molecule. We're also targeting with antisense oligonucleotides for brain disease such as depression and cancer. We're also designing fluorescent probes in our lab in order to search how we can improve the different approaches in a way that we be capable to see it under a microscope. And finally, we're also designing a large-scale library of compounds, small molecules, in order to see how we can find new molecules that will affect diseases. I would like to finish my talk about introducing you the startup biotech that I've been involving in creating right now. And this is the call of biotech is Renesys Bio. And this biotech is basically made from the inspiration of two different Nobel Prize of this year. The one is the Nobel Prize in Physics that have been capable to enable us to use AI and artificial neural networks. And the second Nobel Prize also from this year, and to show you how is it relevant for the days that we are living on, is the structural prediction of biomicromolecules and the Nobel Prize in Chemistry in 2024. So the idea is how we founded the team. So with the team with Dalian Do, and under the supervision of Ion Labs, that the one that are leading the project, and with Yissum collaboration, we are creating a company that's capable to merge AI and in order to identify new small molecules that will bind to RNA that is causing different diseases. So just to recapitulate everything and to tell you what I've been doing. So what we're doing in my lab, and we are very few in the world that doing it, and I'm the only one in Israel. As a young PI, this is very important for me that I'm bringing novelties and stuff that have never been made in Israel. So that's why I went to do my postdoc abroad, to take the knowledge that can be taken and to learn from the best people in the world that are making research. And today I have an interdisciplinary chemical biology lab where we're doing both chemistry and biology. And in order to design new drugs, and the drugs that we're designing are mostly targeting RNA and affecting the RNA, therefore affecting the disease itself. So I hope that I've been capable to convince you today that RNA targeting has an infinite potential, and the sky is the limit. We can have a lot of different new RNA that can bring out and to see how they can affect the disease, and we can think about new way to affect these RNAs that have never been targeted for 100 years, that people have been focusing on only proteins, and now that we know that we can target RNA, the sky is the limit. I would like to finish with the acknowledgements. First, I would like to thank my lab, the lab members, my students involved in the different projects, my lab manager, Judith, postdoc, Deepak, Elias, and Asil, also involved in two different projects. And I would like to give a special thank to my mentor and friends, Professor Matthew Disney. This is many of the research that I've been showing you is the one that I've been involved when I was in the lab of Matt at Scripps Institute during my postdoc. And I've been learning from him the best way to learn the best research is to be with the best people in the world. He's the best one for targeting RNA with small molecule, and I've been learning from him. I'm thankful for this. The funding agencies, Yissum, for organizing the webinar. And if you want to know a bit about myself and the lab, so welcome you to come to my website and to follow me on LinkedIn and Twitter, where I'm posting a lot of different of the research that I'm running on. And finally, if you're coming to Israel, I would like to welcome you and to see the beautiful view from the labs. This is the lab, and this is what I'm seeing every day. So the mountains of Jerusalem, and this is the campus, and this is my students. And I would like to thank you all for joining us today. And if you have any question, I will be happy to answer.
So we do have a number of questions. I hope everybody can hear me.
Yeah.
We have probably 5 or 6 questions, and then a couple additional that I'd like to address. But before I ask the questions, I just want to make one thing clear to everybody. You know, I spoke about the We Are One, but the unbelievable work that Raphael is doing in his lab goes far beyond what Renaissance is. But what Renaissance is came out of the lab. So at the university, he's doing, and he's the only one in Israel doing research in these small molecules. A part of that research gets put over into Yissum, which is the tech transfer group, which is celebrating its 60th year, while the university is in its 100th year, as we all know. And so then the tech transfer group takes that along with the doctor and his what he's doing, and they create a tech transfer entity when possible, and they fund that separately, spin that out with the technology, and fund that separately. And there's many of these, and there's many stories that you've heard of some unbelievable things that have literally changed, you know, the way people have lived, how they've lived, and made their lives better. And this is not just in Israel, it's throughout the world. So what he's doing is really making the world a better place. And so, as he said, that he's got work in his lab. There's Renaissance Bio, and then there's the intersection of that with Yissum. And people can choose to be involved with the lab. They can be involved through funding at Hebrew U. They can look at Renaissance, which is typically more personalized. And then they can look at Yissum, and then also, very important, because this is how the students he gets there, are things like the We Are One campaign. So I just wanted to get to that prior to asking all these important questions. And the first question is, what makes the difference between DNA, RNA, and protein structure?
So that's a great question. And the major difference is between DNA and RNA, we'll start between them, is that the RNA and the DNA, RNA and DNA are made of 4 nucleotides. The DNA is made of 2 different strands that are 2 strands that are complementary one to each other, while the RNA is only 1 strand. In order to be stable, the RNA has to be folded into a 3D way, and this unique 3D folding is the one that's making us possible to have a molecule that will bind there. When we look at protein are using different building blocks that are called amino acids, and these amino acids are making the structure of the protein. So if you think about it, so DNA and protein, you have the RNA that is in the middle way, using the very similar building blocks, but have a very defined 3D structure, similarly to protein and not similarly to DNA.
Next, thank you very much. Next question. How can RNA research help to develop drugs for prevention versus treatment? And today, are vaccines the only way RNA can be used for prevention?
So for prevention, a lot of RNA biomarkers are today under investigation and used in order to identify disease even before you see the appearance in proteins. So there are a lot of biomarkers of RNA. For instance, one that I've been highly interested in is diabetes. So there are a lot of biomarkers of microRNA that are located in in your mouse that can be collected and have very specific uh, pattern that will be capable for us to use as a prevention tool. So you use the RNA that is in your mouse in order to identify the disease even before you know that you have the disease. So there are a lot of research and investigation in this way in order to prevent using RNA biomarkers before the disease has even its phenotype and has been expressed inside the patient.
Is Roger Kornberg doing work in that with the swabbing of the mouth and looking at through RNA?
No, he's using the protein. I'm using the RNA. He's using the protein that's inside the mouse, exactly. So that's the same idea, but now we're using the RNA.
Okay, so you're looking at detection through a swab or a saliva type thing.
Exactly. So you use the saliva that you have in your mouth, and you have very specific pattern for different diseases that can be collected from the mouth. And basically you can see that these different biomarkers can be used in order to investigate and to prevent the disease to be expressed.
Next question, thanks. Next question, does the RNA folding pattern depend on the cytoplasmic environment, things like pH, intracellular pressure, et cetera?
Yes. The environment is affecting the RNA folding. Usually the folding of the RNA inside the nucleus and inside the cytoplasm is quite similar. You don't have a lot of difference for the same RNA in terms of folding. But if you look at RNA outside of the cells, take it in vitro in a tube, you will see that RNA can be folded and unfold using different pH. Also, by increasing the temperature, you will see that the RNA is going to be unfolding. And like all the different biomarker molecules, as well as proteins, are also affected by pH and different temperature and environmental conditions. So as well, the RNAs can be affected in the folding.
One question which you don't have to answer. This unbelievably, I can answer this. Is there a summary of the presentation? And the answer is there is a summary of the presentation. And if it's okay with Dr. Benhamou, you can contact either Matt Cohen or Judith Shankman, and they can help you with that. Another question, which I can't answer. In MYC, how selectivity towards MYC, mRNA, and not another RNA is being validated, and what happens in RNA sequence?
So in all of the different research, I didn't go through inside and deeply inside all the different research that we've been carrying. But we are trying to always measure the selectivity. So the way that we're measuring the selectivity is by measuring by RNA-seq and to see how we're affecting only the RNA that we are targeting. And this is a key experiment in every time that we are synthesizing a new molecule. We would like to see that we are affecting only the target that is related to this. So the RNA target is related to this. And in this case, in the CMYC, we have been capable by RNA-seq to see that in cells, we have not been affecting any other genes, or the genes that have been affected by CMYC are related actually to CMYC itself, so the translation of CMYC itself. So, but we've shown by RNA-seq that our compound have been capable to be highly selective.
What about muscles affected, degraded later in life by having had polio?
I cannot answer because I don't have the answer for this question. It's going to be more a question for medicine for patients, and I don't know exactly how they're going to be affected actually.
Okay. If ncRNA is regulating mRNA levels, why go after the ncRNA and not go after the mRNA?
Okay, so that's a great question actually. So we are going after both the non-coding RNA and the messenger RNA. However, the non-coding RNA, such as the long non-coding RNA, have a very 3D fold that is unique. And this 3D fold has been linked to the non-coding RNA to a lot of different diseases and a lot of different messenger RNA that is affecting. And we are pursuing both non-coding RNA because it's known today that it's related to disease. And when you affect the expression of the non-coding RNA, you actually alleviate all the disease phenotype. If it's cancer or aging and so on, you have many different diseases. Cardiovascular disease is working wonderfully as well. But we are not pursuing only non-coding RNA, as I said. We also did for messenger RNA. And I think that we have the different opportunities. We just need time and investment and the way how we can do it in order to push and to design new drugs to the clinics.
The second part to that is, are there naturally MIRs to the ncRNA?
Yeah, microRNA are naturally expressed inside all the cells. Our cells are expressing more than 3,000 non-coding microRNA. And when you see a disease, you will see a change in the regulation of these short microRNA. And this lack of balance is the one that we would like to correct and to fix. So we will have to see that we can have to regulate back what was the microRNA expression in healthy cells, in healthy patients compared to the disease that have the disease.
Can the small molecule temporarily inhibit the RNA without cleaving it?
Yeah, definitely. We have the two approaches. We can either binding and then just inhibit the RNA processing, but we also can cleave it with the different approach called RiboTac. We have also different approaches, not only RiboTac to cleave today, but we can either have a molecule to bind and inhibit the RNA processing and not cleaving it, or cleaving it when we want to think about scissors that will cleave specifically an RNA that is causing the disease. So that's what we would like to have also.
What about homologous RNAs? Can the small molecule be synthesized to bind several homologous RNAs?
Yes, but we are looking to be selective. So usually what we are looking for is the molecules that are capable to bind only one specific RNA that is linked to the disease. What you're mentioning right now is basically having a compound that will affect different RNA, and this is not what we're looking for. We would prefer to have a compound that will binding and affecting only the specific target. Think about a sniper, but in a microscopic way. So we would like to see how we can affect only the RNA that is causing the disease, but not the different homologous RNA.
How can the target molecules be applied to animals and not only to cells? How do you target them only to cancer cells?
So in vivo studies are under investigation and ongoing for different projects. I've not been capable to show you today, but many of the studies that we have been conducting have been tested also in vivo studies, so in mouse, to see how the compound is capable to affect the disease. The major advantage of our approach is basically that we're mostly doing oral delivery of the compounds, and we don't need any injection through the blood or IVC injection. And that's the most advantage that we have, the best advantage that we have from small molecules, that we don't need any delivery way in order to target and to reach the tissues that we would like to target. It's not related only to cancer. It's related to all different diseases. And of course, this is under investigation. A lot of new companies now are reaching out and contacting me and others in order to advise them and to see how we can help them to have small molecule targeting RNA, because there are a lot of people who would like to know, because of the new FDA-approved molecule that are targeting RNA specifically. So they want to see how. And today it's known because we have now few FDA-approved and also in the clinic stage and different stage 1 and stage 2, more small molecules affecting RNA and that are tested in humans.
If the RNA folding changes with cytoplasmic conditions, how can you predict the RNA folding based on its sequence only? Will it change in disease states when the cytoplasm changes? By the way, this is not your average webinar. This is class. And thank you so much for doing this. And to all the people who I can't see on here, thank you for these incredible questions.
So as that's a great question. We have ways today to measure the folding of RNA inside the cells. So we have wet lab experiments that we can do in order to validate that the RNA folding is indeed what we predicted with the bioinformatic tools. And we can do it in cells, and we can actually then extract the RNA and see how the RNA has been folded inside the cells. So we are doing it as well in order to see how the RNA is folding inside the cytoplasm. Most of the RNA that we are targeting is not only in the cytoplasm, but in the nucleus. And we have seen that the RNA, even though it's in disease cell lines, and you have few different perturbations in the cytoplasmic environment, most of the RNA are still folding in the same way. And when we are targeting a mutation, so the mutation is the mutation or the roots of the RNA, that's what we are targeting exactly. And we're capable to be sure that the folding of the RNA is present there, and then we can target with a small molecule before we do all the investigation, how we can elevate and the disease cell phenotype.
Are you trying the small molecules on patient-derived tumor organoids?
Yes, we are doing it as well. We are growing organoids in our lab currently for cancer purposes, and we are checking the environment and how the molecule are capable to affect the organoids and the cancer inside organoids. This is actually a very great way to introduce before we're doing in vivo studies. So to see how we can have a 3D environment that can be affected, affected by the small molecule, and how the molecule can be able to have the uptake and the permeability to reach the RNA inside the cells, even in the organoid form.
Can I ask a separate question now? If you took a tumor organoid from a patient and could you take it to something like a tissue dynamics and replicate that tumor organoid and try many different things on it to try and kill it?
Yeah, so what we are doing, we are growing the organoid. So we have the cancer cells from a patient, and we are growing the organoids in our incubators. So we have organoids that are growing inside the incubator. So we're making different organoids that we have. And then when we have the organoids, basically 2 weeks to grow them, 2 to 3 weeks. And then after growing them, we are testing on the organoids the effect of our small molecule approach.
Okay, we have 1 minute left and we have a number of different questions, and I don't mean to not ask all of them. Could this therapeutic approach also help with Huntington's disease to engineer molecules that target the detective mRNA before it contributes to the defective protein that will accumulate in the cells and cause the disease?
Yes, definitely. The Huntington disease is caused because of a mutation expansion called it's a CAG mutation expansion that is causing the Huntington disease, and there are few companies actually that are targeting this mutation in their RNA levels. We are not currently investigating that, but definitely there are a lot of promising approaches, and the way we can think about it is basically the same way that we have targeted the ALS. So we have the mutation and you target only the mutation, and if you can cut this mutation specifically and degrade the RNA that is causing the Huntington disease, then you will see the disease to be elevated.
Before ending, I'd like to because we have one more that I just like to, get out, which is how do you make sure that the molecules won't be affected by the conditions of the body, like acidity?
We are not sure. That's why we are testing it. Every time that we have a molecule, we're testing the stability of the molecule. And one advantage of the small molecule approach is that these molecules are more stable. But definitely we checking the bioavailability of the different molecules and the stability inside cells and also in vivo studies.
So there were several questions about how to contact Yissum. You can go on the website, just type into Google Yissum, and you can go on and see different types of research. If you need help navigating it, you can send me an email, and I can help point you in the right direction. Or you can just contact directly to Raphael if you have interest in this or in Renaissance. First of all, in closing, what I'd like to do is for everybody to remember some of the things I said about We Are One in the labs and Yissum, to thank you so much for participating in this amazing presentation, and to thank you, Dr. Benhamou, for taking the time to do this and going as in-depth as you have, and given the time to answer literally every question that's been asked. So thank you all very much.
Thank you very much all for coming. It's been a pleasure.